Thank you so much. So I'm Dr. Dainis. I am a geneticist, and I'm excited to be talking to you tonight about a slightly different kind of genome modification or body modification. So I want to start off with a question. I'm going to give you no context. Do you think right now that you can edit your own genome? Yes. Raise of hands if yes. Oh, all right, okay. Would you? Oh, okay, we have no hands raised. Good call. So I'm going to tell you a couple things about human genome editing, and I want to walk through sort of the answers to those questions, because to the first one, we both can and can't edit your genome right now, and I want to tell you why. So this is Victoria Gray. Victoria Gray is actually one of the very first people to have her genome edited in a clinical trial. So she suffers from sickle cell anemia, which is an absolutely debilitating disease. It causes many of your red blood cells to become sickle-shaped, like you can see down on the bottom there, causes incredible pain crises, and is caused by a single small mutation in the genome. So what a number of doctors and scientists did is they took out a bunch of Victoria's stem cells, they modified the genome in them to correct this mutation, and then put them back in, and now her body creates normal donut-shaped red blood cells. And this was incredibly successful. This has really changed her life. It's reduced these pain crises. It's allowed her to lead a normal, active life, and was lauded by the medical community as a huge success. On the other hand, right around Thanksgiving of 2018, news broke that there were two young twins who had just been born in China who had been edited by this scientist, He Jiankui, as embryos. So what he did is he took embryos that were destined for IVF, and he tried, and did not do so well, to modify a piece of their genome in both of those embryos such that the resulting children would be resistant to HIV. Now, he did this completely in secret. He did not tell the parents all of the potential risks of doing this. He forged a bunch of documents and did this completely in secret. And when the data came out, it became clear that not only did he not make the mutations he had originally intended to in the right way, he may have made other mutations he didn't intend to that we don't know the potential ramifications of. So instead of being lauded as a medical success, he spent 3 years in jail. So, what is different between these two? I want to talk about the differences, and it does go beyond just forging documents and, lying, basically. There's more to it than that scientifically. So we're going to talk about three things. We're going to talk about what is genome editing. We're going to talk about the difference between editing an adult and editing an embryo. And we're going to talk about why you should care, because I'm going to argue that every single person in this room, from the scientists to the comedians to the bartender, need to care about this because it's going to affect all of us. So one of the things you have to understand is that there are a few reasons why this is coming up now, and we couldn't do this before. We needed two things to be able to do this effectively. One is we need a simple way to actually edit DNA, and the other is that we need a way to actually get whatever that tool is to your genome to edit your DNA. Now, the first one, you may have heard an announcement about a Nobel Prize a couple years ago. So in 2020, these two women, Jennifer Doudna and Emmanuelle Charpentier, won the Nobel Prize for their involvement in the development of CRISPR-Cas9. We're not going to go over the details of how it works, but just understand that it is essentially a pair of molecular scissors. So we now have a tool in the lab that we can use to precisely cut a piece of DNA, wherever we want in your genome. And we can do this in humans, in bacteria, in fish, in basically anything we want to. This has revolutionized how we do scientific research because it can do one of two things. If you go into a cell and you cut the genome, the first thing that can happen is that your cell can see that cut and it can repair it incorrectly. It breaks that gene. And this is great for research because if you break a gene in a cell, you can then see what happens and see what that gene might have been involved in. But this can also be important in some types of diseases where potentially if you can shut down the activity of a gene that's causing the disease, you could then be able to stop that disease. The other thing that could happen is you can go in and you can cut that DNA and you can give the cell something to replace it with. So if you have a mutation in your DNA, you can then give the cell something to, without that mutation to replace it with, and in that way, potentially be able to reduce that disease. However, in some small cases, this can sometimes cut in the wrong place. So this is what happened in the case of those CRISPR twins, where sometimes there were cuts in the wrong place. These can cause potential new mutations, and truly day by day there are new scientific papers where we are getting better and better at reducing these off-target cuts, but they do still happen sometimes. So the first thing that we needed was that tool to be able to cut DNA. Second thing was we needed to be able to create some sort of tool to get that to your cells. Now, some of these you might have actually heard of in the news in the past couple of years. These tools have really recently been developed, including things like viral vectors and lipid nanoparticles, which are some of the ways that we were actually able to create COVID-19 vaccines as well. It's another way of delivering genetic material into your cells. But now we have these, and so really in the past 5 years or so, we've reached a point where we have both of these tools to be able to do genetic modification. One thing I want to point out, though, is that your genome, when we talk about your genome, this isn't singular. I think sometimes we think about it as if somewhere deep inside of us we have our one copy of our genome, but that's not true. We actually have about 37 trillion cells inside of us, and with the exception of the red blood cells, each one of those cells has its own copy of your human genome. So if you're going to try and modify it, you need to hit about 7 trillion cells inside your body. That's incredibly complicated. It's as if you were trying to mail out ballots to everyone in the US, but you had 18,000 United States. It is a huge number of cells you're trying to hit. So because of that, it's incredibly complicated. Now, each of you should probably have 2 tickets in your pocket. One of them is purple, and one of them is yellow. Now, these are 2 copies of some gene in your genome, and right now, we're all a tissue. You're each individual cells. And the purple copy, very healthy, wonderful, doing the thing it's supposed to do. The yellow copy, unfortunately, means that you glow in the dark. Not particularly great. So what we want to do is we want to try and deliver some sort of gene editing tool to each of our cells, each of you, to be able to actually correct those yellow mutations. So Dylan is going to be our gene editing tool. And if somebody has a phone with a timer on it, I'm going to need about a 20-second timer.
What?
And so Dylan is going to try and get to every cell in the tissue and modify that genome by giving you a correct ticket.
Wait, do they all have yellow ones?
Yeah.
Okay.
Yeah, no, I know. This is why it's hard. And so, do we have 20 seconds? And so, please try and modify genomes.
Okay, go, Dylan.
So, yeah, we're swapping out purple for yellow.
Wait,
We're going to
what
see
happens?
how many purple for yellow you can do.
Thank you.
And the problem here is that you have to
Thank
actually
you.
get through the tissue. This is a physical problem where you're trying to swap things out, you're trying to cut, you're trying to get to a lot of places.
But wait, what if you've been drinking?
Yeah, that also makes it hard, right? Actually, we'll talk about the liver in just a second. So we're getting, okay, that's it. That's it.
No!
No! So who here, yeah, I know. So who here still has a yellow ticket? Yeah, okay, yeah, most of us. So it's very hard to physically get to all the cells because there's only so many Dylans that we can, inject into this tissue at a time. There's only so many of those gene editing tools that we can get out there. So it's incredibly complicated to do. So what this means is that there are a couple of easy targets for genome editing in your body, and I heard you all get squeamish about the eye before, so I'm so sorry that one of the easy targets is the eye, because we can literally put a needle in it and inject the genome editing tools directly into it. And your eye cells are also very receptive, they want to take stuff up, so very easy to do. The liver, everything cycles through your liver, right? So if it gets into your blood, it's going to cycle through your liver, so you can get stuff to there pretty easily. And blood cells, like in the case of Victoria Gray, are also actually kind of easy to get to because we are pretty used to doing things like bone marrow transplants for cancer, so we already sort of have the setup to be able to take out all of somebody's bone stem cells and replace them again. But everything else is really hard. So if you think about something like muscular dystrophy, which is, again, another debilitating disease that often kills children quite young, it has a pretty well-defined genetic mutation. If we could go in and change that genetic mutation, we could save a lot of these children's lives. But you have to get it to every single muscle cell, including things like the diaphragm, including a lot of internal muscles. And that is just incredibly hard, and we don't have the technology to do that yet. So I want to come back again to one more thing before we talk about what makes these two different and why He Jiankui decided to do editing at the embryo. And it's because inside each of us, there are two different types of cells. There are germline cells, so these are basically sperm, eggs, and sort of the other associated cells around that, and then somatic cells, which is everything else. So what he did in editing these two embryos is he injected this gene editing tool at the very early stage in the embryo when it's 2 cells, 4 cells, 8 cells, because it's a lot easier to get to each one of 8 than it is to get to each one of 7 trillion, right, when you're an adult. So that's why he did this. And so he was able to do this injection, or actually he wasn't. He did it quite poorly and it didn't work. But in theory, the idea was that you could do this injection here and then every single copy of your genome would be edited. In contrast, if we're looking at some of the adult clinical trials that are happening right now, again, Dylan couldn't run through this entire crowd very quickly. If you give an injection, you can get to some of those easy targets, but it's very hard. You're not going to actually have this gene editing tool diffuse through your entire body. If we could actually solve a lot more diseases, but that's just not how it works. And the biggest difference between these two is that if you do germline editing, then that individual's sperm and eggs will also have the mutation and be passed down to their children and their children and their children. So you are not just editing that one individual, you are editing them and all of their progeny. Whereas with somatic editing, because you're not targeting those cells, you're not going to get that sort of generational cascade of mutations. So what this means is that at least as early as 2017, there were couples who were trying to conceive who were offered the chance to edit the genomes of their potential children, right? They were going through IVF, they were offered this chance, which means I think that it is not hard to imagine that any of us could be offered this chance in the next 5-10 years. Technologically, we're getting very close. So we have to start thinking as a society about what is okay to edit and what is not okay to edit. So on one end of the scale, you have things like muscular dystrophy, which I talked about before, an incredibly debilitating disease that kills children quite young. If we could take away that mutation at that embryo level, we could then take away this horrible disease from those children. On the other hand, you have designer babies. Would you edit your child's eye color? Would you edit their hair color? Would you make them taller? These are things that are, again, incredibly complicated genetically. Things like muscular dystrophy are one change. Things like eye color and height are hundreds of changes that you would have to make in the genome. But there's gonna be a gray area in there, and it's gonna be up to literally all of us to decide where we draw the line between what is acceptable and what is not acceptable. Because at the moment, there is no line. There are recommendations from the scientific community, but there really aren't rules or regulations for this. We are catching up and we're doing it way too slowly. So we all as a society need to be thinking about and talking about where we want to draw these lines, what we think is okay, and why. So again, this is the argument I'm going to make to you of why you should care. And again, there are a lot of caveats we can't get to in 15 minutes, but this is something that I truly think is going to be affecting all of us. It's going to be conversations we all need to be having. And so I hope we can have some of them in the Q&A. That is my thought.